Alright, now with all these tools and concepts introduced, the metabolism of the first nutrient I want us to discuss is carbohydrates, and to get us started, I want to build a little overview to guide our discussion. The principal carbohydrate that we will discuss in this section is glucose, and it is located right here on the metabolic map. As a reminder of what we talked about previously, glucose can undergo catabolic and anabolic reactions. Anabolic reactions typically serve to build nutrients and store energy, and on the other hand, catabolic reactions typically serve to degrade nutrients and make energy. So, if we imagine that the cell wants to obtain some energy, the catabolic pathway to degrade glucose is called glycolysis, and you can see on the left the numerous reactions it takes to do so. The final product of glycolysis is 2 molecules of pyruvate, and along the way, it will require 2 ATP and 2 NAD+ to go through with the pathway. In addition to the pyruvate, 4 ATPs and 2 molecules of NADH are produced. Beyond glucose, cells can also use other sugars such as fructose, galactose, and mannose to send into glycolysis to produce energy. We will later see how their pathways integrate with glycolysis. For the moment, pyruvate will be the catabolic endpoint of this section, but you will see in the next section that this pyruvate can be used to generate even more energy in the mitochondria. One aspect of pyruvate that we will talk about here is the homolactic fermentation reaction that produces lactate. This pathway isn't really catabolic or anabolic, but it will be important to talk about as it regenerates the NAD+ in glycolysis. Under certain conditions, such as in low oxygen, this pathway happens very often in our muscles. Another important aspect that we will talk about with respect to pyruvate is that it can be used as the starting point to make glucose via the anabolic process of gluconeogenesis. This is something that mostly happens in the liver, and it takes 2 pyruvate, 2 NADH, 4 ATPs, and 2 GTPs to make 1 glucose back. Now, if the cell doesn't need to use glucose right now, it can store it in a long chain of glucose residues called glycogen via the anabolic process of glycogen synthesis. This process costs 2 ATP to add 1 unit of glucose to glycogen. If the cell wants to get a glucose residue back from glycogen, it can break down 1 unit via glycogen breakdown to obtain 1 molecule of glucose 6-phosphate, or simply G6P, which, as you can see in the map, ties back directly to glucose. Beyond the individual reaction pathways, you will have likely noticed that there are a bunch of red and green lines, especially here in this zoomed-in figure on glucose and G6P. These lines correspond to mechanisms of regulation the cell has in place to coordinate all these pathways. We will obviously discuss these mechanisms, but this will require to talk about some of the key hormones of the body, such as epinephrine, glucagon, and insulin. I will do my best to provide the sufficient context on the action of the hormones, but be mindful that the picture of how the hormones and the organs interact together will be discussed at a later stage. Here, the focus is really on the pathways themselves. With this being said, let's start by covering the different pathways by first considering glycolysis. Glycolysis is a pathway that is performed in every cell, and as you can see from the map, it happens in the cytosol. The main objective in undergoing this pathway is typically to break down glucose and produce energy. And as a reminder of the net reaction for this pathway, 1 glucose with 2 NAD and 2 ATP produce 2 pyruvate, 2 NADH, and 4 ATP. Now, glycolysis begins with the phosphorylation, or in other words, the addition of a phosphate group to glucose to produce glucose 6-phosphate. As it says in the name, the phosphate is added on the 6th carbon of glucose, and it comes from ATP, which becomes ADP after the reaction. If you recall our discussion on thermodynamics, you might remember that this reaction was the example used to illustrate one of the important purposes of ATP in metabolic reactions. To sum it up, adding a phosphate to glucose, just like that, is unlikely to happen in cells, and that can be seen from the positive change in free energy delta G. However, this reaction can be coupled to the hydrolysis of ATP, which is the removal of a phosphate from ATP with water, here diagrammed in the bottom right. The coupling of ATP hydrolysis to the phosphorylation of glucose allows this reaction to have a net negative change in free energy, which makes this reaction spontaneous and likely to happen in the cell. I would suggest keeping this idea of thermodynamics in mind, as we will later come back to that aspect when considering how the entire pathway is regulated. Now, the enzyme that mediates this reaction is called hexokinase and is expressed in all cells, which partly explains how come glycolysis happens everywhere. For your information, I want to mention that there is another enzyme by the name of glucokinase that can catalyze this reaction, but it is only expressed in a few tissues such as the liver and the pancreas. Now, beyond the simple fact of making G6P, this reaction has a few implications in the cell, and to understand them, we need to consider how glucose is transported into the cell. So, it turns out that glucose gets imported into the cell via passive transporters, here shown in green, but these transporters are only specific to glucose, so when the cell produces G6P, that essentially traps the glucose inside because G6P cannot cross the transporters. Additionally, with more glucose being converted to G6P, it effectively reduces the amount of glucose inside the cell and increases its concentration gradient to be imported. This effectively allows more glucose to be taken up into the cell. Now, following the formation of glucose 6-phosphate, the enzyme phosphoglucose isomerase rearranges the structure of G6P to produce fructose 6-phosphate. The rearrangement causes the ring to be shortened by one carbon, and it occurs between the first and second carbon of G6P, here shown by the red arrows. Fructose 6-phosphate then gets phosphorylated again on its first carbon by ATP to yield fructose 1,6-bisphosphate. The enzyme mediating this reaction is phosphofructokinase 1, or simply PFK1. As a note for later, out of all the steps in glycolysis, I would suggest keeping this one in mind because it is the rate-limiting step, and as such, a lot of regulation happens here. Now, the F16P that was made in step 3 can then be broken into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by the enzyme aldolase. It can be a bit hard to see where the breakage happens under this form of fructose, so we can consider the chain form. The chemical mechanism of the breakage is a little complex, but the bottom line is that the cleavage happens between carbons 3 and 4, and thus the part in blue becomes GAP and the part in red becomes DHAP. Between DHAP and GAP, only GAP continues along glycolysis, so DHAP must be converted to GAP. This reaction is mediated by triose phosphate isomerase, and it essentially swaps the ketone group in DHAP to an aldehyde in GAP. Before we move on, I want to mention very quickly that this fifth reaction concludes the first phase of glycolysis, namely the energy investment phase. In this phase, we started with one glucose molecule, spent 2 ATPs, and got 2 molecules of GAP. In the following phase, called the energy recovery phase, we will produce the 2 molecules of pyruvate along with 4 ATPs and 2 NADH. So, picking up with our 2 molecules of GAP, the enzyme GAP dehydrogenase adds a second phosphate to GAP to produce 1,3-biphosphoglycerate. To do so, the reaction requires NAD+ to grab the hydrogen indicated in blue to allow the integration of phosphate. By this process, NAD becomes reduced to NADH and allows the oxidation of GAP. If you are a bit uncertain about the reduced and oxidized terminology, we covered it a little bit earlier in the previous section while discussing the principles of metabolism. Now, moving on to reaction 7, here one of the phosphate groups of 1,3-BPG is used to phosphorylate ADP to ATP, and that produces 3-phosphoglycerate. The enzyme that catalyzes the reaction is phosphoglycerate kinase, and keep in mind that since we have 2 molecules of 1,3-BPG, we produce 2 molecules of ATP back. In the next reaction, the 2 molecules of 3-PG undergo a mutation mediated by the enzyme phosphoglyceromutase to produce 2-phosphoglycerate. As you can see, the mutation moves the phosphate group from the third to the second carbon. Moving on, 2-PG gets dehydrated, or in other words, loses water via the removal of an alcohol group to produce phosphoenol pyruvate. This reaction is mediated by enolase. Finally, the last reaction of glycolysis is mediated by pyruvate kinase and essentially uses the remaining phosphate group on PEP to phosphorylate ADP to ATP. This reaction leaves us with 2 molecules of pyruvate. So, to quickly recap what we covered thus far, here is the summarized glycolysis pathway. The pathway was made out of 2 phases. an energy investment phase where we spend 2 ATPs to charge up the glucose, and an energy recovery phase in which we made 4 ATPs back, yielding a net 2 ATP per glucose consumed. Now, with the pathways covered, I want to discuss how it is regulated. After all, there needs to be a way to shut off glycolysis if the cells don't need to produce energy. You will likely have noticed in the diagram and even when we went through the reactions that there are some reactions that have violet arrows and some others gray. This is because the violet arrows represent irreversible reactions and the gray arrows represent reversible reactions. To understand this terminology, we need to consider again the values of delta G for the glycolysis pathway. Keep in mind that this is a table that we already introduced when discussing thermodynamics in the context of metabolism. Now, as you can see from this table, there are 3 irreversible reactions in glycolysis, one mediated by hexokinase, PFK1, and pyruvate kinase. All the other reactions are reversible, which means that if, for example, we consider the reaction catalyzed by NLase, the direction of the reaction will be mostly governed by the amounts of reactants and products. If there is a ton of 2PG, the reaction will go forward and PEP will be made, and inversely, if there is a lot more PEP, then NLase will run in reverse and make 2PG. These reversible reactions directly contrast the purple irreversible reactions as they essentially go only one way. Hexokinase will always make G6P regardless of the amount of glucose. PFK1 will always make F16P, and the same thing for pyruvate kinase. Because these irreversible reactions only go one way, they need to be regulated to prevent overconsumption of a given nutrient. You will notice as well in the zoomed figures that there is a bunch of green and red arrows that go to the enzymes. The red arrows represent inhibitory mechanisms, and the green arrows represent stimulatory mechanisms. The 2 key mechanisms to regulate these proteins are via allosteric and covalent mechanisms. Allosteric control happened as a result of a substrate binding to the enzyme, which caused its activity to be modulated. Covalent modifications also have an impact on the activity of the enzyme, and the typical example is phosphorylation. Here again, inhibition and stimulation is possible. With this being said, let's now explore in more detail how these 3 key steps of glycolysis are regulated, starting with hexokinase. So, recall that hexokinase mediates the first step of glycolysis as it phosphorylates glucose to make G6P. Here, the primary mechanism of regulation is feedback allosteric inhibition by G6P. So, excess G6P that builds up in the cell will bind to hexokinase and inhibit its activity. This point of regulation is intuitive because if we consider back the role of G6P in carbohydrate metabolism, it's not explicitly shown here, but G6P is the starting point to store glucose and glycogen. So if you have so much G6P that it doesn't get stored in glycogen, then there is no point in accumulating more of it and consuming glucose that might be useful for other cells. As such, shutting hexokinase down will prevent wasting away glucose. Now, the next control point to consider is at the PFK1 reaction, and to cover its regulatory mechanisms, we need a little bit more space. Alright, so remember that PFK1 catalyzes the phosphorylation of F6P to F16P. This reaction is universally considered to be the rate-limiting and committing step of glycolysis, and this aspect is heavily reflected by the amount of regulation PFK1 has. Nonetheless, if we consider the purpose of glycolysis, which is to degrade glucose and make energy, all the regulatory mechanisms somewhat fall in 2 categories. Those who stimulate glycolysis because we need energy, and those who inhibit the pathway because the cell already has enough of it. So, starting with our first modulator, PFK1 gets allosterically inhibited by the presence of ATP. This inhibition is a little puzzling at first, since ATP is part of the reaction that PFK1 catalyzes, so how could it be both a substrate and an inhibitor? To understand this, let's consider the structure of PFK1 from the literature. So first off, what you see on the left is the entire structure of PFK1, and the important aspect here is that it is a tetramer, or in other words, it has 4 subunits. On the right, you can see a close-up on one of these subunits. Now, what is interesting to us is this plot in E. To give a little context on this figure, the experimenters of this research made multiple mutations to a subunit and then measured the different kinetics to see the impacts of these mutations. This aspect is not too important for us, and what I want to show here is that at lower concentrations of ATP, it acts as a substrate and the kinetics of PFK1 are high, but as the concentrations of ATP go up, now it becomes an inhibitor and PFK1 activity slows down. The reason for this shift in enzymatic activity is because PFK1 has low affinity inhibitory binding sites to ATP, so when ATP accumulates, which indicates an excess of energy, the extra ATP can bind to the inhibitory sites of PFK1. Conversely to this, PFK1 gets allosterically stimulated by AMP. Recall that AMP is simply ATP with two less phosphate groups. Since the energy in ATP comes from the loss or hydrolysis of its phosphate groups, you can interpret AMP to be an indicator of low energy as it represents a depleted pool of ATP. Hence, it makes sense that AMP stimulates PFK1 to produce energy. Now, related to AMP and ATP, there is an important reaction to mention, namely the conversion of two ADPs to ATP and AMP mediated by adenylate kinase, here abbreviated as AK. This reaction is particularly relevant to glycolysis because, as you can see on the left from the thin white arrow, the two required phosphorylations at the start of the pathway generate ADP, so these two ADPs can combine to recycle one ATP and produce one AMP that will stimulate glycolysis to produce even more ATPs. Besides these two important modulators, there is also citrate that has an inhibitory effect on PFK1. If you recall this figure, you will see in the panel D that the activity of PFK1 decreases as the concentration of citrate increases. Without going into too much detail here, citrate is the starting point for fatty acid synthesis, which is a process that happens under high amounts of energy. As such, this inhibition is intuitive as the cell does not want to produce more energy while it already tries to store the excess in fat. The next modulator to cover is a very important one as it is a key target for hormonal regulation, which we'll briefly touch on shortly. This modulator in question is fructose 2,6-bisphosphate, which is produced by the phosphorylation of fructose 6-phosphate by PFK2. F26P can also return to F6P via the dephosphorylation mediated by fructose bisphosphatase 2. Now, the F26P that is produced by PFK2 is a strong stimulator of PFK1, and it doesn't interfere with glycolysis in any other way. So, as far as this pathway goes, F26P is simply made to boost the activity of PFK1. The simple way to interpret the modulation here is that basically F26P is an indicator of F6P accumulation, and as such, it will stimulate PFK1 to get rid of the F6P and keep the pathway flowing. But in my opinion, F26P's influence is best appreciated when considering the hormonal control of glycolysis. In terms of the allosteric control, AMP, ATP, citrate, and F26P are the main ones that are considered for that aspect. Now, let's talk about the hormonal control. As I mentioned at the beginning of the carbohydrate section, we will not go into too much detail with the hormones right now because we will have a section later on that discusses their properties in more detail and how they are integrated in the human body. As such, here I want to present an overview of their consequences on glycolysis, but keep in mind that the details about their pathways and physiological roles will be covered later. An important detail to consider for this regulation is that structurally speaking, PFK2 and FBPase2 are two domains of the same enzyme that operates as a dimer. This directly contrasts with PFK1, which only had a kinase activity. Knowing this about the structure, the important takeaway is that the effects of a given hormone on the PFK2-FBPase2 complex will have a dual effect of shutting one domain down and stimulating the other. For example, if we consider the hormone insulin, one of the primary roles of this hormone is to remove glucose from the blood and increase its uptake in the cells. Consequently, the downstream effects of insulin stimulate the PFK2 domain, inhibit the FBPase2 domain, and thus increase the production of F26P, which stimulates glycolysis. With more glucose being processed by glycolysis, more glucose can be taken up by the cells and removed from the blood. In a similar fashion to insulin, another important hormone by the name of epinephrine is an important signal for muscles to signal them to use the glucose and produce energy to supply muscle contractions. As such, epinephrine stimulates PFK2 and promotes the production of F26P as well as glycolysis. The third important hormone to metabolism that we can consider is glucagon. This hormone is often seen as the antagonistic hormone to insulin, and that is because it promotes the release of glucose in the blood. One way it can do so is via the process of gluconeogenesis, which, if you recall our model of carbohydrate metabolism, gluconeogenesis is essentially the process by which cells take pyruvate and reform glucose. We will cover in more detail shortly the reactions that take place to make gluconeogenesis happen, but the bottom line here with glycolysis is that glucagon will inhibit the formation of F26P by inhibiting PFK2 and thus promoting the formation of glucose, not its degradation. Now, as far as PFK1 regulation goes, these are the most common components that regulate it. I know that this regulation can be a bit complicated to understand at first, but make sure you keep the purpose of glycolysis in mind. It should make all the modulators intuitive to understand. Finally, moving on to our third and final irreversible step, we have the production of pyruvate via the formation of ATP by pyruvate kinase. Here again, the regulatory steps can be divided in a need for energy or we have enough, let's put the brakes on glycolysis. Just like for PFK1, ATP again here allosterically inhibits pyruvate kinase as it indicates that there is plenty of energy in the cell. In some reports, PEP has been mentioned to be an allosteric stimulator of pyruvate kinase, but keep in mind that not all resources mention that. Another allosteric stimulator of pyruvate kinase is F16P, and the logic behind this is that since it is the product of PFK1, it signals a need for glycolysis to be stimulated and avoid the upstream substrates from accumulating. Finally, glucagon also has inhibitory effects on pyruvate kinase, and this makes sense since the goal of glucagon is to remake glucose. Again, we will see shortly in more detail how gluconeogenesis happens. Alright, going back to our model of carbohydrate metabolism, now that we have covered glycolysis and its regulation, we are ready to discuss the fermentation of pyruvate to lactate. Thank you for watching this video. If there was anything unclear or there was a mistake somewhere in the video, make sure to let me know in the comment section. If you enjoyed this video and found it useful, you can consider leaving a like and subscribing to support the channel. Thank you again for watching, and I'll see you in our next discussion.